Chapter 11, Regulation of Gene Expression. So cells control gene expression in order to avoid energy waste. How do cells accomplish this? Well, keep in mind that bacteria are unicellular organisms. They need to be selective when they express some genes over others. Cells don't require all of their gene products at all times. Constitutive genes, these are genes that are constantly on or constantly expressed. You would expect these genes to be genes associated with structure of bacteria or perhaps routine metabolism of nutrients that they constantly encounter. Inducible genes, these are genes that are only required at particular times. You might expect bacteria to express some genes, for instance, when they acquire a very rare or specific nutrient. They may want to express a particular enzyme to break up that unique nutrient that they don't always encounter. So basic control of gene expression can take place during transcription, translation, or even after translation. Now, the way that this is accomplished is DNA is transcribed by RNA polymerase into RNA. RNA is then translated into a protein, and then this protein could be modified. So regulation can occur at each and every single one of these states. RNA polymerase could be prevented from attaching to DNA. Sometimes RNA polymerase can transcribe DNA into RNA, however, that RNA may be degraded, or that RNA may form a secondary structure that's not yet accessible. Sometimes transcription occurs, and then translation occurs. However, these proteins, they may have some type of functional group attached to them, so they're not quite functional, at least yet. So there's so many different ways that bacteria could regulate these gene products that they produce, and each of them has their advantages and their disadvantages. Let's go through a couple. So allosteric inhibition is where inhibitory molecules bind to an enzyme, causing a conformation change. And now that enzyme no longer works, and that enzyme can no longer bind to the substrate. Because that substrate no longer binds to the active site, that enzyme will no longer be functional, at least at that point in time. Allosteric inhibition is often the result of the end of some type of pathway, where that enzyme is needed to produce a particular product, but once that product is produced in a high enough concentration, that enzyme is no longer needed. So that product itself, the enzyme produces, that could actually shut down another enzyme involved in that pathway. And we'll talk about this with the trip operon a little bit later. Covalent modification can also alter enzyme conformations. And if this alters the enzyme configuration, this can also alter enzyme activity. And this could either speed up or slow down enzyme activity. Examples of covalent modification: phosphorylation. This is a very common example, where these proteins are produced and then they're phosphorylated. Proteins can be produced and then they could be acetylated. They could be methylated, and they could even be glycosylated, where carbohydrate residues are added to them. And each of these functional groups, these could have a different effect on these proteins. These could affect proteins involved with chemotaxis. These could act as almost pressing a pause button on a protein, where that protein is produced. However, because this functional group is temporarily there, that functional group, that could really need to be removed before that protein is activated. In other instances, these functional groups might be that last missing piece they need in order to become functional. So now let's talk about how bacteria could regulate gene expression at the transcriptional level. So, although cells are capable of modifying enzymes and therefore altering enzyme activity, the energy input to make the enzymes is already expended. This wouldn't be very efficient. A better means of energy conservation would be to only make enzymes as needed. Much of the control mechanisms work to prevent transcription and thus translation. So when genes aren't required, they're not expressed, and that would save energy. An operon is one way that this can be accomplished. An operon is a set of adjacent genes that have related functions, and they're transcribed and translated together. This results in polycistronic mRNA. This is mRNA that specifies more than one gene. Now, to think about this, polycistronic mRNA, this is where DNA is transcribed, but when DNA is transcribed, there could be 1, 2, 3, maybe even more genes on one RNA transcript. Now, that's important, and that's a feature that bacteria use. So when bacteria have multiple genes on one mRNA transcript, that could be very efficient in expressing genes together as a suite of genes. So expression of an operon, they could be regulated at a single promoter, and that's going to help increase efficiency. So at one promoter, multiple genes that are transcribed into polycistronic mRNA, they could be regulated at just that one promoter. So an operon contains structural genes and their regulatory elements. They include a promoter. This is something we've covered before. We're familiar with promoters. Promoters are sequences of DNA that are attracted, or I should say that RNA polymerase is attracted to. In other words, promoters promote gene expression because RNA polymerase has an affinity to the promoter. An operator controls RNA polymerase. As we've discussed in a previous lecture, we had learned that repressor proteins can bind to operator sequences. That could ultimately affect whether or not RNA polymerase can transcribe. A set of genes that work together. These are genes that all have a related function. And a regulatory gene outside of the operon that codes for a repressor. This repressor binds to an operator, repressing transcription. And then lastly, an effector molecule. This effector molecule simply affects the repressor protein. It could activate the repressor protein, or it could deactivate the repressor protein. Effector molecules which interfere with the repressor are called inducers. And inducers increase or encourage transcription. Effector molecules that activate a repressor. These effector molecules, they're going to decrease transcription. They're going to prevent gene expression from occurring. So let's take a look at this example of regulating gene expression with the lac operon. Now, before I get into the lac operon, it's important that I introduce a concept. The lac operon is ultimately a set of genes, and here they are right here, LAC-Z, LAC-Y, and LAC-A, a set of genes that are used to take up and metabolize lactose. So essentially, the purpose of the lac operon is to take up the disaccharide lactose and break it up and use it as a nutrient source. There's a problem, however. And bacteria, well, lactose isn't their favorite nutrient. As a matter of fact, lactose would not be preferred at all. Glucose is bacteria's favorite nutrient, not lactose. So bacteria, they only want to express these genes to take up lactose and break up lactose if, number one, lactose is available. It doesn't make sense for bacteria to exert all of this energy and materials to make these enzymes and proteins if there is no lactose. That would be a huge waste. They only want to make these if lactose is there in the first place. Secondly, bacteria would only make these if there is lactose and there is no glucose. So these genes are expressed only if lactose is present and the primary carbon source. So only if lactose is present, not glucose. If lactose is absent, the repressor protein binds to the operator site. If lactose is present, it enters the cell and binds to the repressor. This releases the repressor and transcription proceeds. So in this instance, here we have LAC-I as the regulatory gene that expresses into the LAC-I repressor. Here we have the promoter. This is where RNA polymerase would normally bind to. Here we have the operator. The operator is the area the repressor would bind to if that repressor is activated. And then here we have the LAC genes. And these LAC genes produce proteins that take up and metabolize lactose only if lactose is available. Well, let's go through these scenarios. Let's say that lactose is unavailable. There is no lactose. If there is no lactose, would bacteria express these genes? Well, the answer, of course, is no. Bacteria don't want to express these genes if there is no lactose. That would be very wasteful. So LAC-I binds to the operator. RNA polymerase cannot attach to the promoter and proceed. So RNA polymerase cannot transcribe genes. In this instance, that repressor binds to the operator, preventing transcription. Let's take a look at another scenario. Let's say that the bacteria did encounter lactose. Well, if they did encounter lactose, this could be isomerized into allolactose. Allolactose is just another form of lactose, but if we look at this molecule right here, allolactose, a form of lactose, interacts with this LAC-I repressor. And look, upon interaction, that changes its configuration. Because its configuration is changed, it no longer binds to the operator. RNA polymerase binds to the promoter. LAC-I cannot bind to the operator. So the inducer, allolactose from lactose, this induces transcription because allolactose deactivates the repressor protein. Now that this cannot bind to the operator, RNA polymerase can bind to the promoter and it can transcribe. Here we have our lactose genes that have just been produced. So here we have some of these genes that are expressed. Permease. Permease is produced by the LAC-Y gene. We have beta-galactosidase. This helps break up lactose into glucose and galactose. And LAC-A. This makes an enzyme called transacyltase. It's not known precisely what the function is. However, there's some research that suggests it helps keep lactose concentrations at optimal levels inside the cell. So now let's add one other layer of complexity to the LAC operon. Remember, in this instance, we had said lactose genes are expressed if lactose is present. The presence of lactose is going to isomerize into allolactose, deactivate the repressor, so the presence of lactose enables gene expression. What about if there is glucose and lactose together? Well, the LAC operon can use an activator protein called the CAMP receptor protein, or CRP. And in addition to the CRP protein, there is a co-activator cyclic AMP protein. And the cyclic molecule co-activator cyclic CAMP, these can interact together. This could enhance RNA polymerase to the activator binding site. The activator and co-activator together increase the affinity of RNA polymerase to their promoters. CAMP concentrations are highest when glucose is low. Therefore, lactose catabolism occurs when lactose is available and glucose levels are low. In this instance, to the left, we have high amounts of glucose. Well, with high amounts of glucose, this would not have cyclic AMP. There would only be the CRP activator, but this needs cyclic AMP or CAMP to be active. It's not active. So what happens is the affinity for RNA to the polymerase to the promoter isn't very high. And because it's not very high, it's not directed or finds the promoter very readily. When glucose levels are low, this CAMP is released. This yellow molecule is cyclic AMP, simply abbreviated as CAMP. This molecule CAMP interacts with a CRP protein, activating this complex. This then binds to an activator binding site, and RNA polymerase is encouraged or directed to the promoter. And notice, there is no repressor protein at the operator. In this instance, glucose is low. CAMP levels are high. This complex is activated. This recruits RNA polymerase. And there is no repressor protein preventing expression of lactose genes. Let's take a look at an animation demonstrating what occurs with the lac operon.
How do regulatory proteins control transcription? An operon allows a cell to coordinately regulate the production of several polypeptides. This image shows the simultaneous transcription and translation of the lac operon, which encodes genes involved in lactose uptake and metabolism. Glucose is a preferred nutrient source over lactose.
Bear with me one moment.
Regulatory proteins interact with a common promoter and operator to control transcription of the operon's 3 structural genes, LAC-Z, How do regulatory proteins control transcription? An operon allows a cell to coordinately regulate the production of several polypeptides. This image shows the simultaneous transcription and translation of the lac operon, which encodes genes involved in lactose uptake and metabolism. Glucose is a preferred nutrient source over lactose. Regulatory proteins interact with a common promoter and operator to control transcription of the operon's 3 structural genes, LAC-Z, LAC-Y, and LAC-A. Transcription of the lac operon is subject to both negative control, as regulatory molecules decrease transcription, and positive control, as regulatory molecules increase transcription. We will look first at negative control, which occurs in the absence of lactose. The gene encoding the LAC-I repressor is located upstream of the operon promoter and operator. In the absence of lactose, LAC-I binds the operator, blocking the promoter and preventing RNA polymerase binding. No transcription occurs. In the presence of lactose, the repressor is inactivated by an inducer. This inducer is allolactose, a metabolite of lactose. Allolactose binds the LAC-I repressor and prevents it from binding the operator. RNA polymerase can access the promoter. Transcription occurs. Now, let's look at positive control of the lac operon, which occurs in the absence of glucose. Even in the absence of the LAC-I repressor, RNA polymerase requires an activator to bind the lac operon promoter. This activator is known as CAMP receptor protein, or CRP. CRP only binds the activator binding site in the presence of its coactivator, CAMP. In the absence of glucose, CAMP levels are high. CAMP binds CRP. The complex binds the activator binding site, increasing the affinity of RNA polymerase for the promoter. Transcription proceeds. In the presence of glucose, positive control of transcription does not occur. CAMP levels are low, so CRP cannot bind the activator binding site. RNA polymerase cannot bind the promoter. Transcription of genes for lactose metabolism does not occur. The combination of positive and negative control of transcription enables E. coli to respond to concentrations of both glucose and lactose in the environment. If the concentration of glucose is low, the concentration of CAMP is high. Activator binds the activator binding site. If lactose concentration is high, allolactose inactivates the repressor. Transcription occurs. If lactose concentration is low, the repressor remains active. Transcription does not occur. If glucose concentration is high, the concentration of CAMP is low. The activator does not bind, even if the concentration of lactose is also high, and the repressor is inactive. If glucose concentration is high and lactose concentration is low, the repressor binds and transcription does not occur.
Okay. Now let's talk about one more operon. Rather than being induced by the presence of a molecule like the lac operon, that's an inducible operon, let's take a look at a repressible operon. A repressible operon ultimately is where a product that's produced at the end of the pathway is going to repress further expression of genes involved in this operon. So a TRP operon is a repressible operon that controls tryptophan anabolism. Now, when you hear tryptophan anabolism, a couple of things to keep in mind. First, tryptophan is one of the 20 amino acids. So this is a building block to make proteins in general. Bacteria absolutely need tryptophan. And if they don't have it, they need to make their own. So tryptophan anabolism, anabolic means building. So this is ultimately bacteria building or making their own tryptophan because they don't have any. They need that tryptophan to make any other protein in that bacterial cell. So tryptophan is one of the 20 amino acids used for constructing proteins. And when levels of tryptophan are low, there are 6 functional genes that are expressed and involved in a metabolic pathway to synthesize tryptophan. In E. coli, the genes used to synthesize tryptophan are found in the TRP operon, which produces tryptophan only when these levels are low. And that makes sense. Bacteria wouldn't want to synthesize tryptophan if tryptophan's already in a high concentration. They only want to express the Bacteria only want to express the enzymes needed to express, or I should say, in order to make tryptophan when there is no tryptophan. They don't want to waste energy making all of these enzymes for tryptophan synthesis if there's plenty of it. Only when there's none of it would bacteria make the protein machinery to synthesize tryptophan. So in this instance, we have a TRP repressor that is normally inactive. That repressor protein binds to the operator, but it does so only when a co-repressor is present. Tryptophan acts as the co-repressor. This tryptophan amino acid interacts with this repressor. When the co-repressor tryptophan interacts with the repressor, now this can bind to the operator sequence. So therefore, when tryptophan levels are low, the TRP operon is active. When TRP levels are high, the TRP operon is repressed. Here, because the repressor protein is not bound at the operator, this demonstrates that tryptophan levels are low. Bacteria need to have their RNA polymerase bind to the promoter and express these TRP genes so they could begin to synthesize their own tryptophan. Now, what happens if bacteria produces plenty of tryptophan? Well, it's no longer needed. So with all of this tryptophan present, this can then interact with the repressor. Rather than deactivating it, this co-repressor activates the repressor. The repressor can now bind at the operator. And when this binds at the operator sequence, this is now going to block RNA polymerase. RNA polymerase cannot attach to the promoter and proceed with transcription because this is blocking it. The only way that it's blocking it is tryptophan acts as a co-repressor to the repressor, blocking transcription. So in this instance, TRP levels are high, and that's why this is considered a repressible operon, because the presence of tryptophan represses gene expression of these TRP genes. Now let's take a look at an animation of the TRP operon.
Proteins involved in the biosynthesis. The TRP operon of E. coli includes a promoter, an operator, and 5 structural genes encoding proteins involved in the biosynthesis of tryptophan. The TRP operon is a repressible operon, meaning that the genes are usually transcribed. When excess tryptophan is available to the cell, the operon is repressed, ending the production of the proteins of tryptophan biosynthesis. The E. coli chromosome contains a regulatory gene near the TRP operon. This gene is constantly transcribed and translated to produce repressor proteins. However, these repressor proteins are inactive unless excess tryptophan is available. Once activated by tryptophan, the repressor proteins can bind to the TRP operator, preventing RNA polymerase from moving beyond the promoter. When a repressor protein is bound, the genes for tryptophan biosynthesis cannot be transcribed into mRNA. When more tryptophan is available to the cell than the cell can use, some tryptophan molecules bind to and activate the repressor proteins. The repressor proteins bind to the operator, preventing the transcription of the structural genes that encode the proteins of tryptophan biosynthesis. This regulation prevents the cell from producing excess tryptophan when tryptophan is already available. When excess tryptophan is not available, the repressor proteins are inactive. The structural genes of the TRP operon are transcribed and translated, producing the proteins necessary for tryptophan biosynthesis.
So I would encourage you to view these a number of times. And if you have questions, please don't hesitate to reach out. Repressible operons and inducible operons, they do take some practice to go through and understand these pathways. So again, go through these animations again. Check out this video again. Ask questions if you have any. And later we're going to discuss how regulating gene expression not only can occur with 3 genes at a time, with 5 or 6 genes at a time, it can also occur with dozens and dozens of genes at a time. And this can have a profound effect on bacteria. And we'll discuss that in our next lecture.